Great pleasure and a real privilege to introduce today Howard Berg to the physics community and some of the chemistry community, as I see here. Howard started his degree at Caltech, his undergraduate work studies at Caltech, where he actually started as a physics student, but then he faithfully met Linus Pauling, and he was persuaded to actually change his degree from physics to chemistry. And so eventually he graduated as in chemistry from Caltech. At the end of that period, he then went to Copenhagen as a Fulbright Fellow to the celebrated Carlsberg Laboratory of Protein Function and Structure. And he worked with a man that is actually very well known for people interested in protein folding, Linda Stone Lang, who actually trained many, many important scientists here in the US at the time. At the end of that period, he came back to Harvard and decided to actually become a medical student. And he tells me that he realized within the first 10 minutes of actually entering medical school, he realized that that had been a mistake. And so he eventually he took some useful courses in the medical school at Harvard, but eventually left after 2 years and joined the chemical physics group at Harvard also. And so he worked with Norman Ramsey and became an expert in lasers and lasers. He built hydrogen lasers and graduated as a chemical physicist. After that, he joined the Society of Fellows at Harvard. He was there for 3 years. And at the end of that period, he was offered a position as an assistant professor in the biology department at Harvard. He studied there from 1965 to 1969. And at that point, he decided to move to Colorado, to Boulder, Colorado, where he became interested in the motility of bacteria. And in fact, he built a microscope that allowed scientists for the first time to follow for a long time the movement and the position of bacteria. And out of those studies and those observations came our current paradigm of bacterial motility, which he published in 1972. In 1973, he discovered that the flagellum motor in E. coli actually rotates, and he became very interested in that. He published that in 1973, and a number of other important papers actually came out during that decade, the decade of the 1970s. In 1979, he moved to Caltech, where he was in the faculty from 79 to 1986. And in 1986, he came back to Harvard, where he actually became a professor, actually Herschel Smith Professor of Physics and professor in the Molecular and Cellular Biology Department at Harvard. It's difficult to actually say in just a few words the enormous contributions of Howard Berg to biophysics and to our understanding of bacterial motility. He has written basically the canon in the area. And in fact, he's also perhaps motivated by his own Brownian motility. He's written a very celebrated monograph called Random Walks in Biology, which actually many grad students now use as a standard reference. Howard has been honored by many, many prizes and recognitions. In 1984, he won the Max Delbrück Prize from the American Physical Society together with E.M. Purcell. And he's a member, of course, of the National Academy of Science, a member of the Philosophical Society, and a member of the the Academy of Arts and Sciences, American Academy of Arts and Sciences. And most recently, in 2014, he won the International Sacre Prize of Biophysics. So Howard, it's really great to have you back here at Berkeley, and we are all ears.
Yes, thank you very much, Carlos. Most of what you said was right. is a complicated story, but it just took me a long time to make up my mind on what I wanted to do. Along the way, I learned enough to do cross-field work. You can't hear me? Consider yourself lucky. Okay, I want to say initially some things about mostly E. coli motility. Some history. I can't avoid history because it's such fun. Talk about the bias random walk, which Carlos already mentioned, flagellar rotation. Talk a bit about the cyclic network and about adaptation, and tell you what our more recent interest is in that set of subjects. But then I'm going to add some comic relief, which is the story about gliding bacteria, which is the bacteria we study. Carlos, I thought you were a good experimentalist. Higher? What do you mean higher? You want it higher? But my mouth isn't at the bottom of my chin. Hello? Are you there? Come in, world, please. I told Carlos earlier that his father must have been a tailor. He started adjusting my clothes. Okay. So the history, of course, starts with the discovery of bacteria with Anthony van Leeuwenhoek. It was, I've forgotten the number, the 14th letter he wrote to the Royal Society in 1676. So it's 100 years before our Declaration of Independence. And he studied a bacterium which is thought to be Spirillum volutans. It's a big, beautiful shaped thing. And what it did was zooming across the field of view, and that's what attracted his attention. So it was motility of bacteria that led to the discovery of bacteria. And this is the frontispiece of a book written by Clifford Dobell, who's a British parasitologist who got very much interested in Anthony van Leeuwenhoek. And he went back and learned archaic Dutch and read all his letters and translated them. And sent his findings to the Royal Society, along with what Leeuwenhoek had done. Van Leeuwenhoek wrote his work up in a quarto, big pieces of paper, in what these days looks like India ink. If you go to the Royal Society in London, you can see these papers, and it's like being in a time machine. And they fold them twice and seal them up with red wax, and then write to Henry Oldenburg, Royal Society in London. And Oldenburg was the secretary of the Royal Society, and he translated these things in snippets and put them in the transactions of the Royal Society. That's how the world found out about Van Leeuwenhoek. So this book was written on the 300th anniversary of Van Leeuwenhoek's birth, so it's really quite remarkable. And I recommend it if you don't know enough about Van Leeuwenhoek, to actually read his research work. My favorite early worker, for reasons that will become clear, is Theodor Engelmann. Theodor Wilhelm Engelmann. And he's a German physiologist who went to Holland and got a position in Utrecht. And if you read up here in 1867, he went to work with Donders at Utrecht, where he married his teacher's daughter and finally succeeded him to the chair of physiology. So the moral here for students and postdocs is you want to get ahead in the world, work for someone who has a reasonable daughter or son. Now, it's actually a tragic story because his wife gave birth to twins and died. And he remarried, and his second wife was a pianist, and Engelmann was a cellist. Together they did a lot of work together, but they were friends of Brahms. So there's an interesting correspondence between Engelmann and Brahms. And you've probably heard of the Engelmann Quartet. It's written about in honor of this man. So the history is fun. Modern work began in 1964 in Japan and in the United States in 1965. And the Japanese workers were interested in the structure of bacterial flagella. They worked with salmonella, which was important in the clinic. And you had immunological different types of flagellar filaments, so you could stain them. And there was a collaboration between a biophysicist, Showa Asakura, cell biologist who knew about the electron microscope, Goro Okuchi, and finally a very good bacterial geneticist named Tetsuo Ino. And they did absolutely wonderful experiments where you could heat up salmonella to 60 degrees and the flagella dissolved. You spin out the cell bodies and cool down the suspension, it remains crystal clear. So you have a supersaturated solution of flagellin, which is the protein from which flagella crystallize. And if you add seeds, little pieces of filaments, each seed will give birth to a grown filament, which they study in various ways. Adler pioneered the work on chemotaxis in E. coli, which is mainly how the cells respond to chemical stimuli. And this is a picture from his first paper in 1965, which is a negatively stained image of E. coli. And here you only see 5 flagella, and they're truncated. They're actually much longer. But this is a negatively stained preparation, and you can see the stain is petered out, so you don't see the flagella beyond the limits of the stain. And Adler picked Escherichia coli, which has been called the hydrogen atom of molecular biology, which is a very simple bacterium. It lives in your gut. It's about a micron in diameter, 2 or 3 microns long. It has a 3-layered wall. It's called a gram-negative organism. So you have an outer membrane which has portholes in it called porins. So any molecule, water-soluble molecule smaller than sucrose can get in without any trouble. It has an inner membrane which separates inside from outside, basically. And then in between it has a quasi-rigid gauze-like material called peptidoglycan, which gives the cell its non-spherical shape. So it's the rigid framework of the cell. And attached to that are the flagellum, which we'll talk a lot more about, and pili, which are thinner, straight, non-modal structures. So, of course, it was one of the first bugs sequenced. It has 4,286 genes. And we have maybe only 6 times as many. So this thing is actually quite sophisticated. And it's been fiddling around for several billion years and come up with solutions that still are proving surprising, as you'll hear. So Adler perfected this assay that had been developed by another German worker named Wilhelm Pfeffer, where you take a microscope slide, shown here, and you put a U-shaped spacer on it, which is just a melting point capillary vent. You cover that space with a square cover slip, and then you pipe it in your suspension of bacteria. And then you slide in a thin capillary tube. This is not the scale, which has a few millimeters of some substance. chemoattractiveness you want to study. Okay. So you wait 45 minutes or an hour, and then you pull the tube out, and you wash off the outside. You take the contents, and you serially dilute and plate them. You can count colonies, so you can count the number of bacteria that get into the tube in a certain period of time. So here's my favorite Adler experiment, published in 1969. It's done with two species of E. coli. The left peak is for a mutant that can neither metabolize nor transport galactose, so it gains absolutely no benefit from galactose. The second one is a wild-type cell which can transport and metabolize. That's called galactose plus. And so a mystery here to you might be, why is the mutant that can't utilize galactose 10 times more sensitive in this assay? As you see, you start with this axis on a log scale is the number of bacteria, the concentration of galactose in the capillary tube, and this is in linear scale the number of bugs that get into the tube in the course of the experiment. So, does everyone understand why the mutant is more sensitive than the wild type? You get an extra cookie if you don't. Why is the mutant 10 times more sensitive in this assay than the wild type? Mutants on the left. It can't metabolize the galactose, can't pump it into the cell, can't do anything. Well, the reason is it can't eat the gradient, whereas the wild type consumes the gradient and reduces the magnitude of the stimulus. So it doesn't do as well as the mutant. But the point here is that chemotaxis is a matter of aesthetics rather than material gain. In this experiment, the cell that has no benefit from dealing with this material still loves the taste. And that was very interesting because it meant you could do all kinds of things that you otherwise couldn't do, and got me interested. I got interested, excuse me, is how the bacteria found their way into Adler's capillary tube. So that's why I built this tracking microscope. So here's 30 seconds in the life of a wild-type E. coli, recorded in 3 dimensions. This is just a 2-dimensional projection. In 1972, which is before many of you were born, old data. But this is a span of about 50 microns, X, Y, and Z. And what you can see is the cell decides to go along a fairly straight line and changes its mind, tries another direction, just keeps doing it. And these straight segments we call runs, and they're on average about a second long. The abrupt changes we call temples, they're about a tenth of a second long. Then if we put one of Adler's capillary tubes out here, we found that if by chance the cell is going up the gradient, the life is getting better, it then postpones this event. Favorable runs get longer. If, on the other hand, it finds itself going the wrong way, it doesn't do anything that it wouldn't do anyhow. So I like to say E. coli is an optimist. If life is getting better, enjoy it more. If it's getting worse, don't worry about it. Which is a nice strategy. And it was fun for me because the leading hypothesis in the field before this work was that bacterial responses, behavior responses, were avoidance responses. The cell backs up when life is getting worse rather than keeps going when life is getting better. I seem to be fading out here. I'll keep going. So it's a biased random walk, and it turns out that when the flagellar motors are spinning counterclockwise, the filaments form a coherent bundle which pushes the cell forward. When one or more decide to go the other way, then the cell jumps about, picks a new direction. And when the rotation goes back to counterclockwise, you get another run. So cells run and tumble. Well, then we realized that bacterial flagella don't wave or beat like sperm tails. They actually rotate rigidly. They're spiral wires, not bending machines, which means they're driven at their base by a rotary engine, which means that nature invented the wheel, and that's kind of wild. Thank you, Carlos. Change color. It's gone pink. So the best demonstration of flagellar rotation is really an assay developed later by Silverman and Simon, where you can knock off most of the flagellum cell and you can bolt one of them down to the substrate. And the rotor now, instead of driving the filament, drives the boat. So look at this. This cell is going clockwise, counterclockwise, 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 clockwise. It just switches back and forth at random. And Adler and company found that when cells continue to swim smoothly in a run, they're running their motors counterclockwise longer than they otherwise would. The tumbles result from clockwise rotation. So, but now this means you don't need the tracking microscope. You just wash an attractant medium across the slide, and everybody goes counterclockwise for 4 or 5 minutes. So you can study chemotaxis at the level of single motors, which is. So the motor is a remarkable device. It has about 20 different proteins in it. They all have names. They're named by molecular geneticists. They were all called Fla originally because the defects were non-flagellatory, behaved as if their flagella didn't work. But then the first one you find is Fla A, and next is Fla B, and so on. But they soon had trouble because they ran out of letters of the alphabet, because there are more than 26. So they got renamed. There was a high-level conference of bacterial geneticists, and they got renamed according to where they were on the E. coli genetic map. So Fla became Ghij. I never can remember how to pronounce these things. Flag, Flu, Flea, and Flush. Now, I thought they should be called Fee, Fi, Fo, Fum, and then you could remember them better. But anyway, this is the state of the art. On the right here is an electron micrograph made by David DeRosier, physicist at Brandeis. It's also negatively stained. But you have at the bottom a big ring, which is in the cytoplasm. It's called the C ring. It has various components, mainly Fly F is the plate here, the next component upon which the ring is mounted. And there's Fly F, Fly G, Fly M, and Fly N. Then you have this mounting ring, as I said, you have a drive shaft that goes through a bushing here, which gets the drive shaft through the rigid framework of the cell wall, followed by a flexible coupling, universal joint, which is called a hook. Then you have some adapter proteins. Then you have this enormously long helical filament. And then you have at the far end, you have a cap. And the interesting thing about this thing, it grows at the distal tip. It doesn't grow at the base. And the polymerization occurs below the cap. So there's a pore, 2 nanometer diameter pore runs all the way down this axial structure through which the components have to travel. And material is pumped into that pore by a structure down here, which is a pump driven by a proton motive force. Now, for a very long time, I was wanting to know, does the rotor, does the basic part of this motor spin at the same rate that the external components do? The external components, the hook and the filament, you can put markers on them, or you can watch them under the microscope, and you can see how fast they go, but how fast does the innermost ring, the C ring, spin? So we devised what we thought was a simple experiment that took us 15 years to complete, only a year or so ago. You put into the flagellar ring, the C ring, and we did it in the ring made up of a component called Fly N, which is the innermost one, of fluorophore. So this was an internal GFP type fusion. And you look at the fluorescence in polarized light, and then at a given time, you turn up the intensity of the excitation being by 100,000 or something, and you bleach the fluorophore. Now, if all goes well and it was a perfect experiment, then as the ring continues to rotate, it rings at twice the rotation frequency. Unfortunately, these guys get out of step very quickly, and so it decays very rapidly. So the actual data, just look at this panel here, shows this sinusoidal behavior, but it damps very quickly. So this is 4 milliseconds, full scale here. And people didn't believe this till we did bootstrap statistical analysis. So here's the best collection of data. The average you can see is sinusoidal behavior, and the control looks crazy. But the speed you get for this thing is about 300 hertz at room temperature, and that's how fast the motor goes if it's running close to zero torque. People still have trouble hearing me. I'm speaking loud enough. Maybe I should just hold this silly thing. I told Carlos his father must have been a tailor, but anyhow, maybe I told you that before. So what we found early on as a consequence of this experiment, actually in doing a control, was that you could stain bacterial flagella with fluorescent dyes, and you could really see what they're doing all the way down to the cell body. So here's a freak that has only one filament, and here its motor is reversed and the filament is wrapping up into a different structure called semi-coiled. It keeps turning clockwise, and the semi-coiled thing decays into what's called curly. It doesn't get back to normal until the motor reverses again. So it's a fairly complicated story, and tumbles are more complicated than had been thought. But I'm going to skip this because we're running out of time. But here you can see bacterial flagella in real time. This experiment shows that the filament can change its morphology and then return to the normal morphology and rejoin the bundle. Now, if you look at the cells near the bottom of the preparation, it's absolutely frantic. I don't know what's making this noise. I'm doing something wrong. So look at this. How can you swim if your filaments are changing their morphology on this crazy way? But somehow it works. Oh, I see. I'm not supposed to breathe. Maybe if I fall over, it would be a more interesting seminar. So, flagella are controlled by a chemosensory system that has a whole bunch of different components. And those have been studied in detail, are still being studied. But I want to call your attention to the yellow guy there called Y, key for Chemotaxis Y, is a signaling molecule that when phosphorylated binds to the base of the motor and tells it to go clockwise. But the lifetime of KY left to itself is about 20 seconds, and that's too long, KY phosphate. Because the cell has to make up its mind in fractions of a second. So E. coli has another molecule, an enzyme called Z, which is a phosphatase, that cuts off the phosphate and shortens the lifetime to about 1/10 of a second. So this devise an assay for this molecule based on frets. So if you make KZ a cyan fusion protein and KY a yellow fusion protein, when this complex forms, you can get energy transfer from blue to yellow. And this enables one to measure the activity or the concentration of this signaling molecule in real time. And that led to a lot of interesting stuff. So cells adapt. If you give them an attractant, you get a response, and that response is sudden and can be complete. So the rotational bias here can go over some finite number down to 0, and then in a few minutes, the cells recover. If you take the attractant molecule out again, then you get a spike of increased activity. Honest, guys, I'm not doing anything. Sorry. What am I doing? Breathing? Okay, I'll stop breathing. So this is a complicated story. You can model it. As a physicist, like to model things, and I've had help here with it by Yuhai Tu, who is a theoretical physicist at IBM. But this network has two properties. They're quite remarkable. One is it adapts exactly. So it doesn't matter what the concentration of a lot of the components are. If you give it a stimulus, it'll come back in time to the initial behavior. But the other thing is it does something that's called fold change detection. This comes out of the theory. So here's an experiment done by Tom Shimazu, a former postdoc who's now at Amolf in Amsterdam. By mixing, you can program a stimulus, some complicated thing, and then you can look at the response using the FRET technique. Now you turn the ambient concentration down by a factor of 10, and you construct the stimulus that's 10 times as small, but you get the same result. So now you can get down another factor of 10, or another factor of 10, or another factor of 10, and the stimuli still, the results all turn out to be the same. And I don't really understand that. I think that's remarkable, but it comes out of the math. It's called fold change detection. Now, some years ago, 1986, as it says here, we looked at mutants that couldn't methylate or demethylate the receptors, and this is a mechanism for making temporal comparisons that makes chemotaxis work. So when people normally say that cells adapt, they mean they're talking about this modification, this methylation of the receptor, which occurs on a time scale of seconds. But if you take a mutant that doesn't have the enzymes that do this methylation or demethylation, and you stimulate them, you get a response, but if you wait a few minutes, most of the result goes away. So they are adapting on a slow time scale what's going on. From the FRET technique, you can look at the FRET signal, and it doesn't show this slow adaptation. So this says that whatever's going on occurs downstream of the KYP molecule. So it must be the only thing you got left is the motor. And what's new is it turns out the motor itself remodels in response to the rotation of it. If the KYP concentration is so low that the cells are only spinning counterclockwise, the motor recruits the molecules in the C ring to which QIP normally binds, and therefore it increases its sensitivity. Now this looks crazy, but the problem is that these fluorescent labels bleach. So instead of going up to some new fixed value, it looks like you're getting some biphasic response, but they're just basically going up to, they're adding these fluorescent components. Now, switching is said to be ultra-sensitive. That is, if you plot the clockwise bias, the fraction of time a tethered cell rotates clockwise as a function of the QIP concentration. This was done by Philippe Cluzel with Stan Leiber. You get a very steep response, which has a Hill coefficient of 10. We actually know the real TL coefficient, I may not have time to explain, is actually 25 or so. So this is one of the most ultra-sensitive systems known in biology. So the addition of the proteins to which QIP binds shifts the operating point of the motor to smaller concentrations. So what's happening here is the motor is shifting its operating point to match the output of the chemotaxis system. So this extends the range over which cells can respond. But it doesn't do it on a second time scale, it does it on a minute time scale. Now, if cells adapt to changes in the signal, what about changes in viscous load? Long ago, we did an experiment called the resurrection, where you can take motors that are missing components of the stator, which we call force-generating elements or units, and you use a molecular biology technique to have the cell synthesize more. The motors recover or speed up in a stepwise manner. So, you get linear functions. So this is step 0, step 1, step 2, step 3, step 4. So the motors add pistons, which is why we call this a resurrection. You get up from your bed and walk. What if you suddenly change the viscous load on a cell? What happens then? What happens is the cell adds more force generators. It modifies itself so it can generate higher torque. And you can do these experiments in two ways. You can take a cell with just a stub, a filament, or a hook and add, using an optical trap, a 1 micron ball, and the viscous load goes up by a dramatic factor. Or you can have this cell with a sticky stub, which settles down and starts spinning, tethered. Either way, you'll take my word for it, the speed goes up in a stepwise fashion. So you go from 0 all the way up to 11 pistons. So that's quite remarkable. And one of the things we're worried about now is how can this information, this mechanosensation, be transmitted to the rest of the cell and modify the genetic apparatus? That's a hard question, and I don't really know how yet what the answer might be. But from the initial speed, when you suddenly increase the load, you can learn how many torque generating units drive the motor near 0 load. It's usually 1, sometimes 2. Well, there's other work we've done that's colorful, literally. You can grow bacterial flagella and stain them with a green dye, and then grow them more and stain them with a red dye. And we can study the rate of growth this way, which has been done most successfully by a worker in Germany named Mark. I'll remember the name in a minute. But let me get to the comic relief. I think we need it. There are bacteria that glide over surfaces at speeds of about 2 microns a second, which is very fast on the scale of you're used to at Berkeley, which is maybe 2 microns a minute. Well, years ago, we studied one of these called Cytophaga species U67. It was found in Copenhagen, and it wound up in the strain collection of the State Serum Institute. It was discovered on an ulcer of a frog, but it didn't have anything to do with the pathology, apparently. But they liked its behavior so much, they put it in their strain collection. And we studied this kind of intensively with Rick Lapidus, physicist. But we didn't have the benefit of any genetics. We didn't know anything about the genetics of this organism. And there's a modern variant which could be the same bug called Flavobacterium, johnsoniae, which is studied in Milwaukee by Mark McBride. And one of his graduate students came to the lab, and we started playing with this again. And they'd found that the cell has a molecule they call an adhesin, this stringy thing, 160 microns long, very thin. And you could find these along the surface of gliding bacteria, and it's comprised of this protein called SPRB. SPR stands for spreading. B for the second one they found. And if you label this thing with a fluorescent antibody, you can see spots moving up and down the cell. So the idea would be if the adhesin sticks to the substrate, instead of it's moving up and down the cell, it would still move up and down the cell, but the cell would move then relative to the substrate. That'll become clear as I go on. Now, the postdoc who came from McBride's lab was named Abhishek Srivastava, found if you treat these cells as you do E. coli to break off filaments to tether them, they stop gliding, but you add an anti-sprB antibody and they do this, they spin. And the rotation axis could be anywhere up and down the cell. And the speed and the torque here is comparable to a E. coli flagellar motor. So this says to us that the motion has to be driven by a rotary engine. Now it's a different kind of engine because if you take E. coli and add a viscous agent and increase the load, it slows down. It runs at constant torque. This motor just runs at the same speed. And I don't know how you design a motor that runs at constant speed, but the cell has done it. All the proteins are different than those in E. coli, but they must be homologous somewhere. The motor is part of what's called a type 9 secretion system. E. coli is type 3. So we're dealing with something that's driven ultimately by a rotary motor. Now if you take gold particles and put anti-sprB antibody on them and study them in an evanescent field where the brightness depends on the distance from the glass substrate, you find that they trace out, this is a long cell, they trace out the right-handed spiral. If you study these on a cell of normal length, which is about 6 microns long, so the green spots on the left there are motors labeled by a fusion of a protein called glide-L with YFP. So glide-L is one of the components of this transport system. And glide-L minus mutants don't glide. Now, if you compare the motion of the tracks mapped out by the positions of gold antibody labeled beads, you discover they're close. So if you plot the distribution of distances, you drop a perpendicular from the bead, from the motor to the track, they hug around 100 nanometers. So this leads us to a crazy idea, which is basically a rack and pinion. You have a rotary engine, it has some pinion that it drives, which engage a tread, which is in green here. And the tread moves along a track. And emerging from the tread are these adhesin molecules. So it's a kind of microscopic snowmobile. And we can't think of any other way of solving this puzzle. And the sizes are right, so the distances, the radii, the speeds, the rotation rates all are consistent. So Abhishek made this model that's, I don't like it in one way because it has too few adhesin molecules. If now the adhesin molecule sticks to the external surface, then the cell moves relative to the surface. So how this behaves, whether it continues to glide or not, depends on whether this tail adhesin rips up from the surface or not. If it comes off, then it continues to glide continuously. It's stuck, then what happens is this cell stands on its nose, it pivots. I have to use the laser pointer here. It runs down until the adhesin ends up at the end, and then it flips over and comes back. Flips over, comes back. It just goes back and forth continuously, and you see this. So this is the crazy idea, and this is the comic relief. So the last 15, 17 years, the fluorescent labeling of the flagella was done by a woman who had been working with me for a long time, named Linda Turner. The fretwork was done mainly by Victor Surgic, and also Tom Shimizu. Surgic is now a Max Planck person in Marburg. He's used fret to study everything, every protein you can imagine. So he's made his career on this. Modeling of the signal transduction pathway is mainly UI2, as I said, is IBM and Tom Shimizu. The initial work on motor adaptation was done by a postdoc who's now in China. The work on the response to increased loads was done by a postdoc who's now at Texas A&M. Richard Brantz was in the lab working on this motor remodeling, and now he's a kindergarten teacher in London, so I don't know whether I drove him to that or not. And the heroic graduate student, or postdoc rather, Gabriel Hosu, did the fluorescence bleaching on rotation, that experiment that took 15 years. He hasn't been with me for 15 years, but he picked up the problem. And the gliding motility work, as I said, was done by Abhishek Srivastava. So I'm sorry for the noise, but thank you. No, normally it just keeps going. But it would have to break. And then you'd attach more of the trailing end and just keep doing this.
Have people been able to isolate any of the components of the motor gliding system?
Well, I mentioned there's a whole list of mutants, proteins that have been identified, but no one knows what they are really. Glidel being one. But they're involved in transport of things needed for this machinery. I mean, this is a problem crying for the help of a cryo-EM tomographer. I mean, it's a wonderful problem. All this hardware has to be there. I'll guarantee you, if you study it, you'll find it, become famous. No, it's driven by proton motive force. Right. Yes.
So you showed what happens when you change the concentration of the attractant. So what happens if the attractant comes on and then it's just constant concentration? Does it go back, does the bug stop, or what happens?
No, they keep swimming merrily. It depends what mutant you're. The wild type cell will recover from this in a few seconds, behave as it was initially.
Meaning that it just tumbles right, I mean, it goes
Right back to its original run-tumble behavior, even though the attraction is still there. And this is a consequence of this methylation, of receptor methylation. If you take a mutant that can't do this receptor methylation, that's missing the enzyme, its behavior will persist. In other words, it'll swim more smoothly and just keep doing that. Yes. Well, yes, the motor, in fact, steps. It's been studied in Oxford by, this is named Richard Berg. It has 26 steps per revolution, basically. But to measure that, you have to work at very low torque, because the elastic twist of the tether will filter out any such structure. And so he actually did the experiment with cells that are chimeric. You can run them on sodium ions, or you can run them on protons. The best data you get is with sodium ions, because you have better control over the concentration. I don't know if I answered your question. You had me?
I'm curious how the flagella are coupled, how they can couple together, so if you were to start twisting about the springs together, so they would all make a huge mess. So how they
People have modeled this, chemical engineers who've driven flexible wires in pots of silicone oil, and you see these phenomena. It's just low Reynolds number hydrodynamics. And the fact that, filaments can run into each other. I mean, once you get two filaments behind the cell is pushed forward and the rest gets swept to the back. And then they get intimate with one another, form bundles. I have a wonderful model of two spiral wires you can twist and just go around side by side. So originally I thought, if these things rotate, why don't they get all tangled up? That turns out to be not a problem. Yes, it's helically shaped. That's right. In the opposite direction, it tends to jam. The filaments tangle up and the motor stops. But they're mutants. You can get flagella of different chirality. Yes?
What's it about the landmarks necessary to lay down the spiral track on which the bracket can be run?
Absolutely no idea. Our data say the track is there, and how it gets laid down is a complete mystery. But what it's made of, what proteins. There is some fragmentary EM data coming along that indicates these things are not a complete fiction.